What's the difference between PCR and NGS when it comes to DNA testing for infection?
Welcome to another MicroGenDX Minute.
More and more physicians are moving away from microbiology labs with standard culture testing and are moving toward molecular diagnostics using DNA testing. But is all DNA testing the same? The two most commonly used tests for infectious disease are polymerase chain reaction, also known as PCR, and a more advanced DNA test, next-generation DNA sequencing, known as NGS. PCR compares the DNA in a sample against a panel of microbial species which are believed to be common pathogens in that area of infection. These panels are limited to a predetermined list of species, which represent 1% or fewer of all known microbes. If you're only using PCR, you may be missing out on important diagnostic information. Unlike PCR, MicroGenDX's next-generation DNA sequencing compares the DNA in a sample against our curated database of over 30,000 microbial species to identify all of the species in a sample. In fact, to date, MicroGenDX has identified more than 6,500 different species of bacteria and fungi in human samples. Our NGS test correctly identifies all of the microbes from each sample. The report even breaks down the percentages of the species detected, identifying the most dominant species contributing to the infection. To give physicians a complete diagnostic picture of all the microbes in the infection, MicroGenDX can use PCR and NGS on every sample. To learn more about the difference between PCR and NGS and how a more advanced DNA testing can help you, visit microgendx.com.